Tell me what you remember about Seymour Benzer.
Where should I start? Well, when I went to Caltech, I started working in the Bonner's lab, and then Benzer had just arrived there, and so I was asking him about stuff. And in college, I had some experience working in flies in the genetics lab, and I decided to just do some phototaxis and so forth. And then there were a bunch of people at Caltech that were interested in rhythms. So we had our little rhythm group that would meet like once a month or something and discuss circadian rhythms. And so then I got interested in rhythms and thought about this project to try to look for fly mutants. Because first of all, Benzer had me working on Notch, I think it was, some kind of weird thing. So I was supposed to take the qualifying exam in biochemistry. So we spent the whole summer, a bunch of students formed another study group, and we studied all summer for this exam in the fall. And while I was doing that, I said, well, okay, I'm going to screen mutants all summer, and if I don't get anything, then I'll move on to something else after I take the exam.
That was 1968.
So, yeah, that was 68, that's right, the monumental year when all kinds of stuff happened. Then according to Konopka's law now, as it's in the book, I found a mutant. And I remember presenting it to the Benzer group, and I was very reticent about making claims, but there was an internal control using that attached X technique. So the males were mutagenized and the females weren't. So the females were rhythmic and the first mutant was not. And that's measuring eclosion. And so then I kept screening, and I got per short and per long out of it. Yeah.
So could you explain the attached X in more detail for the film?
Attached X means that the females have two X chromosomes that are physically attached together and a Y chromosome. So the progeny get their X chromosome from the fathers instead of the mothers. And they're expressed because there's XY, males are XY. So the males will express the mutations even if they're recessive on the X chromosome.
And you exposed the males to the mutagen.
Yeah, EMS, right. That was Ed Lewis's technique.
And then, so you were coming in twice a day to check eclosion.
That's right. That's exactly right.
And so could you describe? You would come in the morning and count.
Well, no, actually, I come in at the beginning of the light cycle, which was in an incubator, and at the end of the light cycle. And so there should be a lot more emerging during the day than during the night. So I just shook out the ones that had emerged into an etherizer and just looked at them. And most of the time, at the end of the dark period, there was not much there that emerged in the dark. But then sometimes you get more that, And so those were processed further in these so-called bang boxes, these eclosion monitors, which Hindry was nice enough to send me a few of those.
And you needed those to look in constant darkness?
Yeah, those were in constant darkness for the free running rhythm.
All right. So you got your first arrhythmic mutants, and you presented it to Benzer's lab group?
That's right. Yeah.
And you were reluctant to make any claims, and they got all excited.
Yeah, and Benzer said, well, that's what you want, right? I guess so.
So, so Benzer was not really checking in on you with any regularity? You were off, kind of doing it on your own? And was that typical in his lab? Was he kind of hands-off?
Yeah, I would say so. I mean, he had these technicians who would do stuff that he wanted them to do, and he was isolating these strange behavior mutants, SB103, things like that, and he isolated the drop dead mutant and a bunch of stuff like that. But yeah, I was more or less left to myself. And every so often, like every couple of months or so, you get into some frenzy about something, and then I'd just leave. I just wouldn't show up for a few days until he calmed back down again. Like with Ross Hodgetts, he came in one day and he says, "Well, you have to put labels on all the drawers in your lab, and they have to be exactly this size and exactly, you know, so and so." So Ross had to drop everything and put labels all over in his lab. So every so often you get into a frenzy about something like that, and like I said, I just take off. I go hiking or something to the Sierra Nevada.
So the group that you had discussions about that summer that you were studying did not include Benzer. This was other students.
Hmm?
So you were discussing about rhythms. You said it was the summer you were preparing for your exams.
Oh,
And that was a student group?
The little group?
Yeah.
No, actually, there were a couple students or a few students, but it was Stromwasser. He was the one who was the big rhythm person at Caltech at the time. And then there were some postdocs, people that worked in his lab, and people that worked in other people's labs that were postdocs that were interested. So there weren't very many students in that group.
So you mentioned Jerry Feldman?
Yeah, Feldman did a postdoc in Norm Horowitz's lab, and I was actually assigned to Norm Horowitz as a student to do the rotation in his lab.
On Neurospora.
But
Was that on Neurospora?
on Eurospora, yeah. And so I did a few things there, but then I decided I didn't really care for it. I moved to Bonner's lab, and Horowitz was busy working with JPL on the first Mars investigation into life on Mars, so he was never around. And so then before Feldman got there, I switched to honors group, and Jerry said that he wished I had stuck around and he could have supervised me. But anyway, that's the way it went.
So you got, in your paper, you got 3 mutants from 2000 F1 males.
Right.
And was that surprising? Was that a lot? Was that a little? Was that what you expected?
Well, I was glad to get period mutants as opposed to arrhythmic mutants. So
But first you had the arrhythmic. That was the first one that came out.
I think so. That's all I remember. But still, when I got the 3, I thought, I bet these things are going to map to the same place. And sure enough, they did.
You expected that, really?
I did, yeah.
Why not expect lots of different genes on various chromosomes and maybe several on the X?
Well, I don't know, the phenotype was just so strong. I thought, well, we hit something here. I mean, Benzer was one of the things he was famous for was these hot spots in mutagenizing bacteriophage. I thought, maybe this is a hot spot.
Great. Yeah. So that's amazing. I mean, that per long, per short, and per naught all were so close. But in those days, so you were aware of Boehning and later Pittendrigg showing heritability of period. So you knew there was a genetic basis.
Right.
But how did
the dogma was that it was very polygenic. That there were going to be lots of genes and they all combined to produce this clock. So they didn't think mutagenizing one particular gene was going to do anything except maybe a half an hour difference or something.
So by they,
everybody, Nobel Prize winners, Max Delbruck, anybody who had an opinion about genetics.
and Delbruck was incredulous when he saw the data.
Oh yeah, well he didn't see the data. He said we were sitting around. In fact, this was in an article that I have someplace in the Dallas Morning News, I think it was. They talk about this party at Benzer's, and Benzer says, "Oh, Ron has found these genes that affect the circadian clock," and Delbruck goes, "I don't believe a word of it." That was his standard reply to everything. I don't believe a word of it. So He didn't believe, and so I'm just sinking down, in my chair, toward the floor. Uh, but I don't think he ever did believe it.
And the first time I met you, I remember I was a first year graduate student. There was a meeting in San Diego, and you were presenting, and I remember distinctly you were using a ski pole as a pointer.
Oh yeah, yeah.
But there must have been an occasion before that you first presented it to a circadian audience. Do you remember when you first presented it?
I don't. I really
Any of the
don't.
early presentations and how people responded?
What year was that? Do you know? Was it
73. Would have been 73 or
Oh,
74.
So it was real.
You were
Well,
well.
I was a PhD by then.
Hmm.
Well, the first real meeting I remember is the one that I mentioned that in Maryland, where there were all these big shots from various universities, and it was 2 or 3 days. They're all talking about the clock, and at the end, people agreed that if the clock hit them in the face, they wouldn't recognize it. So how do you even recognize if you have a clock on some subcellular level or cellular level? They didn't even know whether it was cellular. That was while these people did these experiments on, whether you need only one cell or many cells in a multi-celled organism, because they know there were single-celled organisms that had clocks. But the question is, with a more complicated organism, did you need a network of cells or not?
I remember those discussions about emergent properties.
Maryland, yeah.
Okay, we're live again. Right,
And that was when a big earthquake occurred out here in '71.
I don't remember.
And my girlfriend was at Scripps, so I was really worried, but you couldn't possibly get a phone call into this area after that huge earthquake.
Was Pittendrig at that meeting?
He was.
And do you remember his response when he first saw your work?
Well, they didn't have much comment. They didn't know what to make out of it.
But he had already sent you the bang boxes,
Oh yeah, yeah.
so he knew what you're doing.
I had those by '68.
Right. So were you the first person to take an occlusion phenotype and then look at the behavior of individual flies?
I think so, because there was very little in the literature at that time about monitoring activity, and so we had to build our own little activity monitors and get the records like that with individual flies. Yeah, that was pretty important because the arrhythmicity could have still meant that each fly had a clock, but they were all out of phase. There was something wrong there.
The problem still comes up all the time.
Yeah, well, the dogma for the circadian clock was that you measure the circadian clock, the fundamental oscillator, by two properties, the period and the phase. So, I mean, what else have you got? So, but yeah, there were all these ridiculous things that people published, like in the Journal of Theoretical Biology, this one guy published this slipping belt model, where the clock is still going at 24 hours, but it's attached to something else, and the belt is slipping, And every time I would say, where is there a belt in a cell? I mean, this is idiotic. And I had to deal with these moronic comments the whole time.
Right, but in those days, so when you went after and got a locus, what was in your mind about how you would get sequence and get a protein and then figure out how the protein product interacted with some other proteins, presumably, or mechanism?
Yeah, well then the Drosophila cloning didn't exist. So it took some years before the people, especially I think this guy Jerry Rubin, made the right plasmids and the libraries and so forth. So that took quite a while.
But you anticipated that finding the gene
Would give us a protein eventually. Yeah, that's right, because DNA makes RNA makes protein, and people just forget that. They think, oh, maybe it changes the water structure, or maybe, all these absurd, moronic things which I couldn't even deal with. It's like, because they're not geneticists, they forget that DNA makes RNA makes protein.
But so this was like late 60s, and I can't remember what was the state of sequencing once you had a locus and you assumed that it was one gene. But could
Well,
you really know that?
I used this deletion mapping because it was in this well-studied region of the X chromosome, this so-called Zes white region. So there were a lot of deletions and deficiencies and translocations available. So I went through and did a bunch of those to show that in fact they were going to be affecting the same locus essentially.
Your paper, you think Barbara Stewart
Barbara Stewart.
Stewart for mapping experiments? She
Oh.
must have been the technician who.
Oh, she was a grad student, I think. I'm not sure.
So, but what I would like to get some perspective on is, okay, what were your plans once you got the gene for going into the mechanism of how, how do you go from, okay, even if I were to know that there was a deletion and of a base or an inversion or something, what was your thinking about how you were going to go from there to finding a cellular mechanism or even a multicellular mechanism in a fly?
Well, we were working on cloning the locus. Once the technology was there, I had this postdoc, Janine Perlman, who was working in. She went over to Elliot Meyerowitz's lab. He was nice enough to provide some supervision for trying to clone this gene.
He does Arabidopsis.
Well, he did Drosophila first, and then she messed it up basically, and so and then I moved to Clarkson University, and then I had people asking me, calling me up, asking me if they could work in my lab and if they could do some cloning and this and that. But with Clarkson, they just promised all this stuff and they delivered on nothing. They promised an independent biology program, but it never did get independent. And they interviewed a guy who was very qualified, but the dean decided he didn't want to give up his power, so he sent the guy a contract, and then he called him up and said, no, send it back. And so they messed up the whole thing at Clarkson, so I was in no position to do any cloning over there.
So between your graduate work and your coming back to Caltech, you postdoc with Pittendrigg at Stanford. And can you describe the work you were doing there and your interactions with Pitt?
Well, again, Pittendrigg wasn't there half the time because he was on this committee to investigate nepotism or something, which, looking back now, I find bizarre because Stanford is like the most nepotistic place. This guy that I knew here got a job up there, and his wife, he married this girl, and Chinese girl, and she works in his lab. So they're making a fortune, but anyway, Pittendrigg wasn't there all that much.
Were you trying to get mosaics at that time? Because you mentioned in your discussion of the paper that you were taking that approach.
I think most of the mosaic work I did was after that. I'm not sure exactly what I did at Stanford. I know I worked on a collaboration on crickets, trying to localize the clock and look at the optic system and so.
Was that with Terry Page?
No, that was with this guy Phil Sokolov, the guy who devised the chi-squared periodogram, and then another guy who was at Berkeley, a German guy, whose name I forget.
Werner Lohr.
Yeah, Lohr, that's right.
He was on my qualifying exam committee and my thesis committee.
Oh, is that right?
Yeah, he was a good guy. I liked him.
Yeah, he was a pretty good guy, but
Yeah.
but then for some reason the collaboration just dropped off. They couldn't get crickets for a while or something.
So that's really interesting because I know that the Bushel Sokolov. I don't understand the math.
Your light just went on.
Yes, thank you for telling me that. All right. Okay. So,
The light's still off.
this one or this one? That one. Okay.
This one's
This is the backup camera. So
Okay.
just to make sure, this one seems to be going fine though, so I'm not worried. So I would like to just get your recollection. I mean, there's this lore around that Pittendrigg was hostile to genetic approaches to finding out about the mechanism of the clock, which to me as a non-geneticist was kind of crazy because I think he was a Debjanski student and he appreciated genetics. But you didn't encounter hostility.
Well, they were different types of genetics. You know, there was the Dobzhansky was a population geneticist, and so everything was populations and selection and that kind of thing. So that's what Pittendrigg knew. He didn't know anything about that. In fact, the first time I sent him a mutant, they counted all the flies together, the males and the females. And I said, no, you got to count the males separately. So then he did it again and found, agreed that there was a mutant. Well, he was sympathetic. You know, they sent him my first grant application, and he was sympathetic for a little while, but then, he turned against the whole genetic thing. Same way with other people. Like Stromwasser, he was sympathetic for a while, and then he turned against me too, and the whole genetic thing. So.
But what we remember, of course, for Pittendrigg for was this formal analysis and phase response curves.
Right. Yeah.
Was your feeling that doing the genetics would make that irrelevant, or what?
No, descriptive stuff. It's like, developmental biology. I mean, that was 90% descriptive stuff, and so I figured it's the same way. You have to describe things, and I didn't really want to do it, but I probably should have done descriptive stuff for my mutants. Just like dark cycles and this, that, and the other thing. Just to put it out there. But I was having trouble getting stuff published because nobody would publish it. And what finally was, Mel Green came up to me and said, he was on the board of molecular and general genetics, and he said, well, if you can send your papers to me and I'll get them reviewed. So that, otherwise I would have probably had 0 published because there was no, Journal of Chronobiology, whatever. There was no Journal of Neurogenetics. None of that stuff existed.
Do you remember the reviews for your PNAS paper? That was 71. It was published in 71.
That was communicated at the time.
So it didn't have to be reviewed.
Benzer was a member of the Academy, so he didn't have to review it. But that was pulling teeth also. We'd just be going back and forth on some things. He'd write it one way, I'd write it another way, and it was just like pulling teeth. And finally he was supposed to go off to the Salk Institute for the summer, like he always did. And I said, we got to get this thing finished before you leave. So one last push, and then he sent it in, and at least it got published. Otherwise, that wouldn't have gotten published either. Because he had this Delbrookian attitude towards papers. Delbrook said you should publish one paper every 10 years. Yeah.
It's not a survival strategy.
Yeah, well, things were changing. Nothing was set, and even the journal Genetics, they were so slow. If you sent Genetics a paper, it would take like 2 or 3 years to come out, and then they finally changed that. After some years, but there just wasn't a good place to publish this stuff. Science, they give it mixed reviews.
But I'm surprised because, I mean, having published that first grade paper in PNAS, I would have thought you'd have a easy path to subsequent work, subsequent publications.
You mean in PNAS?
The 71 paper.
No, but I mean this.
Not necessarily in PNAS,
not
but I
necessarily.
mean that certainly got everybody's attention, didn't it?
No, because the clock people didn't know genetics people didn't know clocks. My grants, it was the same kind of thing. they put me in the genetics study section, which at NIH, which insulated me from all these crazy clock people that thought that genetics was good for nothing. Uh, so it insulated me from them. But then on the other hand, they didn't always get an ad hoc person who could do it justice. So sometimes I just wouldn't get the priority score I needed to get the grant.
Well, we still face that. I can guarantee you. It's still.
Well, yeah,
still
I'm sure
going
it's
on.
just gotten worse. I mean, Jeff was telling me about some panel that he was on, and it was just nuts the way he described it.
Yeah. So, again, that's the backup camera. I'm not too worried about that. In our phone conversation, you were describing interactions between Pittendrigg and Benzer.
Well, yeah, they finally met for the first time. Pittendrigg was invited to give a talk at Caltech, where he actually said during his talk in front of everyone that he wouldn't hire a person to do Drosophila work.
Pittendrigg said it.
Pittendrigg said that, yeah, in front of the whole Caltech audience,
Because,
because he didn't think it was worth anything.
but yet it was his favorite organism. I mean,
Well, he was referring to the genetics, you know. I mean, he used Pseudo obscura because it had a sharp rhythm, and that was a decision I had to make at the very beginning. Was I going to use the melanogaster with the not so good rhythm, or was I going to use Pseudo obscura with the sharp rhythm? But I decided the genetics was an overpowering reason to use melanogaster. And then Pittendrigg did isolate a couple of arrhythmic strains in Pseudo obscura, but he never did anything with it.
so the early and late strains were Pseudo obscura, not melanogaster. Yeah. So that's but had you done your stuff in Pseudo obscura, you couldn't have done it because you didn't have the map.
Well, yeah, I would have had to invent the genetics of Pseudo obscura by myself, which.
Okay. And then one other question. So for mammal-oriented people like me, it seems like a real stroke of luck that the gene was on the X chromosome. But if you could explain for the film why that's not such a surprise given the number of chromosomes, etc.
Well, the X chromosome has approximately a third of the genes because there's lots of genes on the X, on chromosome 2, chromosome 3, and then chromosome 4 is very small and it doesn't have a lot of genes on it. So you're looking at about roughly 1/3 of the genome. And we actually did look for genes on the autosomes. You can do that. It's a longer procedure. You have to homozygose the chromosomes. But that's how Mike Young found his double time, is it, I think?
Timeless, yeah.
So we tried, but we didn't find anything.
So, given that you were clearly interested in looking at the behavioral rhythm, and in your paper you mentioned that with mosaics it maps to the head, was it your interest to go into the neurobiology?
Yeah, we did some experiments to try to localize it further in the brain, but the mosaicism was such that I couldn't really pin it down too much.
Because I'm also thinking that, given some of Benzer's other work on behavior mutants and learning mutants, that maybe there was this anticipation that it would go into the nervous system.
Well, yeah, sure, yeah, that's what. And then after we did those brain transplant experiments, then we proposed that it was probably had some neurosecretory aspect to it.
Yeah, I kind of remember that. Could you describe a little bit about that, the experiments and what you found?
Well, we put, I think it was short period into arrhythmics, and managed to get short periods out of them.
So
there was some kind of circulating hormone or whatever, that peptide that synchronized the rhythm.
Yeah, and that was. You didn't pursue that to see if there was either any specific chemical agent or if there was any reinnervation. I mean, Terry Page had done the optic lobe experiments.
Yeah.
in the crickets where it requires axonal connection.
Well, we tried eye mutants, but the eye mutants all had clocks. So even the cineoculus, which was a really extreme thing, it had not only did a lot of specimens not have any eyes, any facets, but we were lacking some of the underlying optic lobes also. But cineoculus had rhythms.
Mm-hmm. Okay. So you expected that the adult behavioral rhythms and the occlusion rhythms would be affected by the same mutation.
Mm-hmm.
Was it really, I mean, I guess it was an open question because nobody looked at the behavior before, but just the significance of that.
Yeah, and it was very penetrant also. I mean, you didn't get like some short periods that had 24-hour rhythms, one out of 50, one out of 20. All short periods were short periods, all long periods were long periods. And then, we isolated this andante, which was like 25 and a half hours. And that was a whole bit too, because there was sort of a miniature mutation close to andante, so people were wondering whether miniature was the same as an andante or not.
So, the picture now is that the clock mechanism is expressed in cells throughout the organism, and that was shut off. It could be okay. That would. Whoops, going again. Okay, so you did cell cultures.
Yeah, and I had cell cultures from a short period mutant, and in a light-dark cycle, or no, we used temperature cycles, not light-dark cycles. We used temperature cycles, and in a temperature cycle, the short period mutant was had a different phase, suggesting that in fact the genes were expressed in the cell cultures. But I couldn't really do it long enough to be able to get a good free-running rhythm out of it. And so I sent the results that I had to Nature, and they says, well, we'll publish it if you get the free-running rhythm. And I never could get the free-running rhythm.
So what? Sorry, what was it that you were measuring? How
cell division, numbers of cells.
Yeah. I mean, it's having a reporter that you can monitor in real time is really the trick for all kinds of circadian.
Well, yeah, those fluorescent things.
Yeah. Okay. I'm just trying to think about what we have left out. So I was just kind of interested in. You said on the phone that something like Pittendrigg was surprised that Benzer was such a wuss.
Yeah, that was his comment.
Could you
well,
put that in the context in which you heard it and what he meant?
That was just a single comment that he made.
So he
thought the Benzer would be a much more forceful personality, but the Benzer was always a real meek, meek person. So that's what surprised him. Is my term as was. He didn't say that necessarily, but a quiet, non-forceful person, as opposed to Pittendrigg, who was like a bulldog who'd go after things.
Yeah, I mean, Pittendrigg was very domineering, and I
Yeah,
remember
that's
meetings.
Right. That's exactly right.
I remember meetings. People would give a talk, and then everybody would wait to hear what Pittendrigg thought about it. These were like really smart people.
Yeah, him, and the same thing with Arnold Eskin. He'd be over there in the audience, and he'd be shaking his head and stuff, and people would get rattled during their talk. They'd start hesitating and forget where they were, because Arnold would also strike out at people.
Yeah, Pittendrigg used to sit back and doodle. He used to draw those. And so you remember that in 1977 we had that course in Pacific Grove?
Yeah, I still have a couple of T-shirts from that course.
Yeah, I think I do too. So I remember you and Terry and Jeff Elliott and Gene Block, and you guys were like camp counselors.
That's right. Yeah.
and you had me sorting flies. I was looking for one with a white eye and one with a red eye, and I went through like 1,000 flies and didn't find any. I thought, I'm sticking to hamsters.
Yeah. Yeah. Oh well.
But it was good. What Pittendrigg was doing was that these meetings, there would be people working on all different organisms.
Yeah. No. Those were, he took the initiative, and that first one was the biggest one in '77. That was it was excellent for the field and interest in it and everything. And then he had another one in '78, but it wasn't this big. But yeah, they were good. I'm glad he did it. You were at Berkeley at the time?
I was. I was a graduate student with Irv Zucker. And Benji Rusek was the student ahead of me.
Oh yeah.
Who had left by that point.
Oh, Rusek.
Yeah. All right. Well, I think we've hit a lot of the high spots. Is there anything you have in mind that you think is part of the story that you'd like to leave for posterity, to the extent that anybody's going to watch this?
I can't think of anything.
Okay. Remember the chronon theory? You're talking about the slipping
Just
belt.
real vaguely.
There's an idea that it would take 24 hours to finish transcription,
Is that what it
whatever
was?
the clock gene was. But I remember going to meetings and having this thought about, if you found the core of the clock, that would it oscillate? Would gene expression oscillate? We're looking at autosomes, so you obviously thought there would be other genes that Per would be interacting with.
Mm-hmm. Sure.
And were you thinking of it as a feedback loop, or didn't know?
That was one consideration. Yeah, that there was some kind of feedback involved.
Yeah, it's just so amazing. I mean, this is, the course of our lives, and you look back at, what did I know and when did I know it, and, like it was obvious, or should have been obvious, or how could we have thought that,
I was reading a review of that Time, Love, Memory book. You know that book?
Hmm?
Jeff Hall spent a lot of time with that guy who wrote it. And there was a review of it by this guy. What was his name? I remember the name, but I can't remember what the guy did. Oh, Barandis, I think. Sam Barandis wrote a review of it. And he said that he thought it was completely astonishing and miraculous that I found those mutants. He said it was unbelievable, completely unbelievable that I found those mutants.
Well, it was remarkable.
But it's Konopka's law, If you're going to find it, you're going to find it fairly soon,
Well, but do you really believe that? I mean, do you really believe that? How much should you persevere if you think your idea is really good?
Well, it works. It worked with Takahashi and his mutants.
That's true. That's true.
It works.
That's true.
Oops. Oh.
Just a sec, it's my daughter. Hey, Sarah, can I call you back? I have to. Can I call you back? I'm doing an interview. Sorry? Yeah. Well, it went off. I'll call you back. Bye. So.
What is that music? It's Beethoven?
Yeah. It's second movement of the Moonlight, but I can't get it. I don't know. I have to get my daughter who does the camera has to help me with my cell phone because, like I said, I'm a Luddite. So, but this is as I was saying at the start, that it's remarkable that as a graduate student you were left so much to your own devices, because nowadays, like when I have a graduate student, and if they spend like a week doing something that I'm not really paying attention to and they flounder, I think, oh shit, I'm going to like lose my funding. But then the question that graduate students certainly, but all scientists would face is like, how much should I stick with this? I think it's a great idea. And your idea, well, if it doesn't come up in the first 200 or whatever, forget about it.
Well, if it didn't come up specifically, by the time I took that exam, then I was going to forget about it. So however many that I had a screen, I was screening while I was studying for that exam. So,
Huh.
so I set a time limit on
So
it.
it's that
wasn't a
you don't
number.
necessarily think that you should stop looking if it doesn't come up really fast. It just, but it is a mark of how well your approach will work within the technical abilities you have at the time. That
Yeah,
I
that's
should come
right.
up quickly. I mean, there are so many things that we do that are like at the limits of what we can achieve technically,
Sure, absolutely.
and that's why it takes a long
Absolutely.
time. Yeah, but hindsight is great.
Yeah, that's right. 2020.
Right. Okay, Ron, I think this was great, and so the project will include, going back, to all the early stuff with plant leaf movements, and the like Demoran did this experiment about putting the mimosa in the dark, and the leaves still moved. It's not really clear how he could tell they were moving if the plant was in the dark. And his conclusion was that it was being driven by the sun, but by something other than light. So you can do a brilliant experiment, and it shakes things up, and then you reach the wrong conclusion.
Well, yeah.
So I mean, one of the Pindrig would make this case that he was fighting against Frank Brown, who really thought that there
Yeah,
was
that's right.
exogenous
Sure.
control.
Exactly.
But I've interviewed some of Pittenbrug's students who were in his lab at Princeton in the 50s. Nobody believes that. Do you remember anybody?
Well, what did they think he?
Well, that Brown was laughed off because his hypothesis was so implausible. But arguably one of the accomplishments of finding a gene was that it would later wrest this idea of anything but an endogenous basis.
Well, Frank Brown still did. They put up an experiment on the Skylab, and Frank Brown was supposed to have an experiment up on Skylab too, and I forget what happened, but I know that the one Pittenger put up, well, I think the astronauts screwed it up or something, and they pressed the wrong button or something, and they never did get results out of it.
Well, the one I remember was that they had Neurospora culture, and they wanted to see if the conidiation rhythm would persist, and it didn't when it was in orbit, and it ended up that the gas exchange was not right in weightless conditions. So it's a permissive conditions question.
Yeah, no, this was a Drosophila experiment, and like I said, the astronauts messed it up, pressed the wrong button or something, and they never did get any results.
So I was on a panel reviewing these people who wanted to look at human circadian rhythms in orbit, and I realized there was going to be a real compliance problem because these astronauts, they bust their butt, they train for like a decade to go up, and then they're in orbit and the sun rises every 90 minutes and you're telling them, don't look out the window.
Yeah. Well, yeah, experimental design, that's a big part of it.